Welcome to all. In the previous video, we have discussed about the collection of soil sample in a surface sample and a profile sample. In this video, we are going to learn how to process that particular collected soil sample and the collection of undisturbed soil samples. So, processing of sample follows the particular points. It includes drying, sieving, grinding and mixing and partition and storing. So these are the major three activities will be taking place in processing of soil sample. What are the equipment and material required for the processing of soil sample? We need a drying cabinet, which is totally optional, and auger motor with a rubber pistol. Or instead of this, you can use a mallet, a wooden mallet we can use. And we need a 2 sieve, a scientific sieve is required. Followed by we need a spatula for proper cleaning, and we need a storage jar, or you can store in a very clean polythene bags. And finally we need a logbook to enter all our soil sample details. As continues, very first we will see drying procedure. So drying of soil sample should be done in shade, not in a direct sunlight, where there is a good, it is a good, not god, sorry to disturb, good air circulation facilities available. And some chemical analysis required analysis without drying. Mostly if you're analyzing nitrate, nitrogen or exchangeable ion, we need not to go for drying soil sample. But all our analysis, all our chemical analysis will be expressed in oven dry weight basis. And the next is called grinding. Grinding, or for our convenience we can say malleting. Remove large stones and gravel, stubbles and straw and grass before grinding. Even though we have taken a lot of care while collecting the soil sample, there is a chance of a very few stones and gravels, and sometimes we can see some straw or grass bits. So we have to remove while grinding or malleting. So we have to take in account that if the soil sample contains stones and gravels in a significant amount, for the research purpose, the portion of this fraction of the total soil weight must be taken into account in expression result in the chemical analysis. So this point is totally for a very particular scientific analysis we need. And we have to avoid the practice of discarding large volume of soil aggregates taking a small portion of sieved soil sample. So if aggregates as a major role in your objective means, we have to keep it and we have to avoid that, and we have to keep a small portion of soil sample if aggregates are very important in your analysis. Continuation. Sieving. After doing all the above two processes, we have to go for routine soil testing, other chemical analysis. We have to pass the soil sample in a 2 sieve, because the highest particle size is sand, which is 2 So in soil science, we mostly rely on sand silt portion content, and most of our chemical and physical properties, we mostly rely on these particles. That's why we have to sieve the soil sample in a 2 sieve. Followed by, if soil contains stones and gravel in significant amount, for the research purpose, the portion of this fraction to the soil weight must be taken into account for expression result in chemical analysis. If we want that gravel portion, we can analyze, for example, after passing that entire soil sample in a 2 sieve, if we need that gravel content, we can express that in our chemical analysis. And we have to avoid discarding large volume of soil aggregates. For example, we should not throw the large aggregates, because that aggregates may contain some clay particle or a sand particle or a silt particle. So we should not throw all those large particles while sieving. After sieving, we have to follow the mixing. So after doing all the above three process, we have to mix those samples in a square cloth or a paper roll, or we can go for a polythene sheet also soil. So in a roll, soil from one corner to opposite corner, hold two diagonal corners. That means, for example, you are keeping soil sample in a particular cloth bag, we have to hold one corner, this corner and this corner, and we have to mix it. We have to mix it in a such a way that it should be mixed properly, followed by some process holding other and diagonal corner. So like this we have to mix it and repeat the process for at least 5 times. So this continuous mixing should be done for 5 minutes. For example, we have to, for example, this is the entire soil sample. From here we have to mix once again, catching from this corner to this corner, we have to mix diagonally. So this mixing should be done at least 5 times for getting homogenization. Like this. As I told, we have to spread our soil sample in a polythene sheet or a cloth. Then we have to mix it. After mixing all these things, we have to we will learn all these things. So partitioning or quadrating. After thoroughly mixing, keep the soil sample at the middle of the cloth or a paper in a form of dome. Dome is like this. We have to, after mixing, we have to keep like this, followed by slightly flattening on the top. We have to give some beating and divide the dome into two equal halves with a sharp knife, two or four equal halves, and again divide into two equal halves. So ultimately we will get the four quadrats like this. In this picture you can see like this, we have to divide in a four quadrats. So discard any two quadrats. Like for example, in the first time you can discard this one and this one. So automatically you will make this. So mix other two parts. After discarding, again we have to mix these two. So like this we have to, again we'll get the same portion. Again we have to make it in a similar procedure. All these above points, again we have to repeat. Again we have to make it into four quadrats, and we have to discard, and finally we have to make it at least 500 grams of soil sample left on our on our paper. And finally we have to go for storing. What are those? So after completing processing sample, we have to store the soil sample with the proper labels for a long time and further laboratory reference sample analysis. The full details of the samples must be in a logbook. We have to enter all our details about these samples in a logbook. So finally, we need, sometimes we have to collect the soil sample in a undisturbed manner. That means we have to collect the sample in a core sample we need, mostly for water holding capacity, bulk density, particle density. To analyze bulk density, particle density, for analyzing all the above properties what I have told, we need undisturbed soil samples. So undisturbed soil sample mostly required for, as I told, all this purpose, pore space retention. So we need some equipments. What are those? We need a core sampler, which is very important. The core sampler is that it looks like this. It has a two cylinder. One cylinder will be cover another, and there will be another cylinder will be inside. This is the core sampler is required, followed by spade, which is very quite common, and we need a dagger or a knife. Mostly daggers are very good, or knives we have to use. So these are the materials required for the collecting of undisturbed soil sample. And what is the procedure to be followed for collecting the undisturbed soil sample? With the help of hammer, drive the soil sample in a vertical soil surface slightly enough to fill the sampler, but avoid the compression of soil in the confined space of the sample. So the sample should be very natural and the core should be very intact to the cylinder. Using the spade, dig out the sampler without disturbing the natural position of the soil sample. That means natural position, how the sample looks in a field. We should collect the entire core. We have to collect the entire core. We have for our laboratory analysis. So separate the two cylinders retaining the soil mass undisturbed in the soil sample. Inner core. What is inner core means? In a particular core, in a core sample, there will be two cylinders. This is upper cylinder. Inside there will be another cylinder and covered with the rings. Covered with the rings. What will happen while we are pushing this entire cylinder inside a vertical soil sample? What will happen? The entire core, that entire soil will be reach here, and while we are taking analysis, we have to remove this upper core and these rings, so we will get our entire core. So we have to trim the edges of the sample with the help of knife. Like this, we have to collect the samples, at least 8 to 10 such cores. Desired depends upon our depth, and mostly 8 to 10 cores are suitable for 1 hectare land. Thank you.